Hi, everybody. My name is Shelby Bacus, and I am a graduate student at the University of Alaska Fairbanks. And today I'll be discussing the effects of ocean warming and acidification on the behavior and physiology of an intertidal grazer and a key sea star predator. Now, our oceans are in a current state of change. And what we know is that our oceans are going to both warm and acidify concurrently. And this makes multiple stressor experimentation an emergent area of study, which ultimately aims to characterize and predict the individual and interactive effects of multiple stressors on marine organisms. And this is important because multiple stressors are going to affect both individuals, but also communities and ecosystems. Now, for our experiment, we looked at Laudia scutum and Evasterius trochelli, or the Pacific plate limpet and the mottle star. And we chose these species because they represent a dynamic predator-prey relationship in the Gulf of Alaska. And they are the perfect model to characterize the effects of ocean change on species in the rocky intertidal ecosystem. Now, the objectives for this experiment specifically were to determine the individual and interactive effects of ocean warming and ocean acidification on the thermal tolerance, metabolism, cortisol expression, and anti-predator behavior in Laudia scutum. Now, in order to do this, we ran an experiment for 21 days, and it was fully factorial in its experimental design and consisted of 4 different treatments. Now, over the course of 21 days, we ran a thermal tolerance assay, a metabolism assay. We looked at cortisol expression and the behavior of Laudia when introduced to their sea star predator. Now, our results were quite interesting, and we'll start with thermal tolerance. So on the right-hand side, if we look at this figure, we have CT max, or the critical thermal maximum of Laudia scutum, the temperature at which they expire on the y-axis, and on the x-axis, we have our different treatments. Now, what we found was that temperature had a significant effect on the upper thermal tolerance of Laudia. And what this means is that those acclimated to lower temperatures actually experience a higher CT max compared to those acclimated to the future predicted warmer conditions of our oceans. But what we found is that there wasn't any significant effect of pH on CT max, and there weren't any interactive effects of pH and temperature as well, which was quite interesting. Now, for metabolism, our figure on the left, we have the mass-specific metabolic rate of Laudia on the y-axis and the time or the experiment in days on the x-axis. I also want to point out that we ran preliminary experiments for metabolism on field-caught Laudia, so looking at the natural metabolic rates of Laudia in their environment. But what we found was that over the course of the experiment, metabolic rates actually increased through time. And that by day 14, the metabolic rates of Laudia were actually quite similar to those of field-caught Laudia. And by day 21, the metabolic rates in Laudia were highest in the low pH treatment. So there's effective pH and time on Laudia on day 21. However, there were no significant effect of temperature, and there were no interactive effects of pH and temperature on the metabolic rate of Laudia. Now, for cortisol expression, we measured cortisol expression over time, but we also measured cortisol expression both before and after being exposed to their sea star predator, because that's a stressful condition and cortisol is released as a stress hormone. Now, what we found is that cortisol expression increased over time. So by day 21, cortisol was highest in the high temperature treatment. So there was an effect of both time and temperature on cortisol expression in Laudia. However, there was no effect of pH nor any interactive effect of pH and temperature on cortisol expression. And when we looked at cortisol expression both before and after being introduced to our sea star predator, we found that there wasn't actually any acute difference in cortisol expression, which was also very interesting. Now, for anti-predator behavior, we looked at 2 key behaviors that Laudia expresses in the presence of their sea star predator. And that is either the decision to actively flee away from that sea star predator or to rotate around their visceral mass. Now, at first glance, we thought there might be an effect of pH on behavior. So the decisions to flee or rotate seemed to have decreased in the lower pH conditions. However, when we ran our chi-square analysis, we found that there was no effect of pH or temperature on the anti-predator behavior of Laudia. Now, the implications for the study are pretty vast, but to sum it up, I want to say that, again, our oceans are going to both warm and acidify concurrently, and this means that there can be both individual and interactive effects of OA and OW on individuals, but also communities and ecosystems. So I hope this experiment really drives the need to design multiple stressor experiments that also integrate multiple levels of biological organization and multiple disciplines to better predict how our marine species are going to fare in the future. Now, with that, I just want to say thank you to my advisor, Amanda Kelly, my lab mates, and funding partners. And with that, I'll take any questions. Thank you so much and have a great